Brandon. How are you?
Hey, good. How are you?
Good to see you. Thanks for having me on for this.
Yeah, thanks for joining me. I appreciate it.
Awesome.
Today, I'm joined by Dr. Pranam Chatterjee, a professor of bioengineering and computer and information science at the University of Pennsylvania. He works at the intersection of biology and AI and runs the Programmable Biology Group at Penn, where his team builds new AI models for designing proteins and peptides. This work can lead to new medicines, gene therapies, and even tools for cleaning up the environment. He also works closely with students and has co-founded several biotech companies that translate his research into real-world applications. Pranam, thanks so much for joining me.
Thank you so much, Brandon, for having me on. I'm excited to be here.
I'd love to start with some of the basics. I think basic for you, but maybe not for others. What exactly are proteins and peptides?
Yeah, it's a great question. Biology is so cool, and they already have great tools for us that we can utilize for biotechnology and great applications. So proteins are maybe some of the most complex tools that biology has developed, right? If you think about your high school biology class, we have DNA, and that DNA will become at a certain point into RNA, which is this intermediate molecule that you may have heard of. But the real point of the DNA and the RNA is to make and code for these complex machines that we call proteins. The proteins are made up of 20 pieces. These are amino acids. And those 20 pieces can come together in various ways. You can think about all the combinations of 20 letters that will make a protein. And those proteins can fold in weird shapes. They can do cool things. They can make your body run, essentially. They can go, every organism in the world has proteins that allow them to live, to eat, to survive, and to fight off diseases and other functions. A peptide is just basically a smaller version of a protein. So if a
Mhm.
protein is a set of 100 of these, one of these 20 amino acid, 20 letter combinations, a peptide is like less than 20. So in that case, you can consider a peptide anywhere between maybe 5 of these together or up to maybe 40, but 20 is like your average. The beauty of having them be smaller, as you mentioned, Brandon, is that when we want to go to actual applications, smaller is better. Smaller means I can make them easier because they're
Oh,
smaller.
right.
Smaller also means that I can also, if I take them as a drug, that they are able to do their thing much more easily. So hopefully that gives you a just nice background of what a protein and a peptide are.
Yeah, that's great. So, what are some simple, if you
Yeah,
will, simple examples
sure.
of, I guess, naturally occurring proteins and peptides in the human body?
Yeah, let's take a great example that you're probably familiar with are antibodies, right?
Mm.
We have to fight off diseases. What does that? Well, the first thing that happens is the disease-causing molecule, usually it's a protein itself. What recognizes it? It's an antibody. And an antibody is produced by your immune system to specifically recognize non-self or non-natural or non-you proteins. That's a great example. For a peptide, I think one thing that you guys may be very surprised by is that, Ozempic, right? Wegovy, the weight loss drugs, that's a peptide. It's a
Oh, okay.
short sequence that is able to bind to another protein and activate pathways in the body, activate signaling or activate just events in the body that allow you to be less likely to eat things. So overall, those just are some small examples, but shows you how powerful these molecules can be.
So, you lead the Programmable Biology Group at Penn. What
Yeah.
is programmable biology?
Yeah, so if you think about the proteins and peptides I talked about, if you wanted to develop a new one, it's hard. It may
Yeah.
take you years to say, oh, I want a peptide that doesn't, solve weight loss, but maybe a peptide that cures cancer, or a peptide that, or a protein that can do this new thing. What I
Mm.
like is that I don't want this to be every time we want something new, a new protein, a new peptide, a new molecule to do something cool or to do something impactful, it takes us 10 years to do it. I want something where, regardless of what your function is, regardless of what you want, here's a model. In our case, because I'm a computer scientist, here's an AI model that will generate that molecule. So it's programmable. You press a button, you get the molecule you want.
I
And
see.
therefore, it becomes both. And it's nice because it means two things. It means the programmability of getting what you want, but it also means that we're utilizing computer science to make that happen.
So, first question, are there new proteins and peptides that are being generated in the human body over time? This might be a dumb question. I don't even know.
Oh.
Like, was there a standard set of proteins that we've had forever and that's it? Or over time, are new proteins actually developed by the human body that are identified?
Yeah, answer the question in two ways. One, yes, is most of the time you have a set of genes, right? That is who you are, your
Yeah,
DNA, and those DNA can create only a certain amount of proteins, right? Like that they code for
right.
those set of proteins. However, those proteins can also be modified after they've been made to do
Okay.
different things. So we definitely have some diversity there. Now, our immune system is constantly making new proteins to recognize all the weird things that come into our body since we were
Right.
two when we started eating mud from the grass, or up
Right.
to now when we get a virus. So we are producing new proteins to recognize and to destroy new
Oh,
things that our body sees, but for the most part, we're stuck with a really good and comprehensive set of proteins that can do stuff. Now, if you have a disease, sometimes it's caused by a new protein that your body accidentally makes because your DNA gets mutated.
right.
You make a new protein and that protein does something bad. You want to get rid of that protein. And so I think that there are different ways of generating new proteins that your body does. But our goal, Brandon, is to make sure that if there is a bad protein that we want to get rid of,
Yeah.
design one ourselves that can do that. We
Wow.
want that level of programmability.
So the idea is that you're not only designing new proteins, but that you're enhancing our existing proteins.
Definitely, right? I think that idea of protein design versus protein engineering is very
Yeah.
important. Sometimes biology has already figured out a great solution. We just need to make a better variant of it. And in that
Yeah.
case, we would be optimizing an existing protein either in our body or somewhere else in nature. You may have heard of CRISPR, right?
Right.
Which is the biggest, one of the biggest discoveries of our lifetimes. It's a protein that bacteria naturally made. And we just repurposed it and re-engineered it from a bacteria, put it into a human, and baby KJ that was fixed, a mutation was fixed at Penn, is probably the biggest clinical story of the last decade, was healed. He was healed because of that protein. And so, it's a weird way of saying we may not have to reinvent the wheel. We may be able to take proteins that exist in nature in our bodies, make them better, and have them do what we want them to do.
Okay. So what does the current process of protein design actually look like in
Yeah.
practice? So you start with some input, right? You want to solve some problem, I imagine. It's
Yeah.
not blind, right? You're not blindly coming up with, I guess, new combinations. What's the input and what does the AI actually produce? Or what does the model produce? What's the output?
So you brought up a great question of two types of problems in AI. AI, guys know from ChatGPT and from Gemini and from others, it's a generative model, right? And so there's two types of generative models. There's a model that would be called unconditional generation, meaning just make me something, right?
Right.
And that would be like what you said at first, just make me a protein. I don't know what that protein does, but it's a protein and it does something. It may have some
It
natural
might.
function, right? It may or may not. The
Okay.
second one is like, I want a protein that does X things, right?
Right.
I want to bind or actually interact with a bad protein so that I can get rid of it, right?
Right.
That's the first area of protein design that is huge, is the idea of designing binders. Something that here's a protein, it's a disease-causing molecule, maybe it's a metal that you want to get rid of, whatever, design something that binds to it. That's actually
Mhm.
what my lab has, for the last few years, really focused on, and is making those really tight binders. Now, the other thing is I want to make a protein that can perform some activity. I don't just bind, but I remove that protein. So I want to
Oh,
degrade it. Or
okay.
I want to bind and I want to modify that protein so it's now healthy. And so in that case, we need a class of proteins called enzymes, which are just more machine-like, catalytic. They are just capable of performing a specific function. So protein design primarily is classified as unconditional, which would be just make me a protein. Conditional, that allows me to interact with a protein like a binder.
Okay.
You, like maybe something where what we're working on now, Brandon, is multi-objective protein design, where
Right.
you're
Right.
like, I need a protein that binds, that is not toxic, that is, very active, et cetera, et cetera. And so I think protein design encompasses these three areas. My lab has been I think mostly a big, I would say, pioneer of sorts in these last two areas. And we've been putting out a lot of really exciting models that do that.
Okay. So what is the model? The
Yes.
models that you develop, what do they actually do? Like, let's say it's not unconditional, but like a more targeted use case. I
Yep.
want to bind to a bad protein, right?
Yeah.
And maybe remove it. What is the model actually doing? Is it taking all the information from the thing that you want to target and saying, let's try a million different combinations of what? Molecules, amino acids, all these different things. And then it somehow comes up with some solutions, or this one might work, and then you test it?
Yeah, that's a great question. And I think when you think of generative modeling or generative AI, you have to I think the best analogy is actually to go back to a tool like ChatGPT or
Yeah.
maybe your image generators. And remember, when you prompt ChatGPT, you're basically starting with a sentence, right? You're like saying, give me a happy sentence that describes X. And it'll
Right.
just generate from that. But the thing is, the model is basically generating things based off what is previously seen. So
Yes.
basically a left to right generative model. And the
Yeah.
way ChatGPT and Gemini and all of these models were trained was on just a ton of like sentences. They weren't sentences where they were pairs of function, like, verb and the actual verb, right? It wasn't that. It was just what we would call unconditional generation. But now for protein design, you want to give it an input, and that input could be, in our case, a protein that you want to target. The model needs to know a lot about that protein
Right.
and then be able to produce an output. And so there's two classes of models that exist in protein design. There's models that use structure, which would be the geometric orientation of the protein, like literally the shape of the
Okay.
protein, and then design. And people like, so there's one camp of people that are very much do this. David Baker, who won the Nobel Prize, done really beautiful work with structure-based design. I would say 90% of the field has focused on looking at the geometry of a protein and then designing something that does something to it.
Mhm.
Our approach has always been sequence models. If you think about a language model,
Right.
you never told ChatGPT or Gemini or Claude, this is a noun, this is a verb, this is how the sentence looks. No, you just gave it sentences. And by training on a lot of sentences and text, it learned the properties of grammar, of syntax, of semantics. We think the same thing is true in protein. If I gave it a lot of proteins,
Oh,
right,
right.
then you should learn properties of those proteins without having to go through that intermediate, which would be the structure of the molecule. And so what we say is give it the sequence of the target protein
Okay.
and give it the function that you want that molecule to have generate. So this idea of conditional generation is really important. And I think my, dogmatic, maybe controversial statement in this is that I really don't think we need to go through an intermediate visualization of the protein to get all of its information. And so
I
for
see.
us, we take in the sequence and we are able to, and maybe other objectives like properties of the sequence, and then generate a new one that would be optimized or one that would actually be useful for an application, like a binder or an enzyme.
Ideally, does it give you like one solution or one potential
Ah,
solution? Or do you specify, I'm going to simplify it. I want 10 potential solutions
yeah.
and we're going to test them all.
Yes, 100%. So generative models are beautiful. If you prompt Gemini with the same question three times, you don't get the same answer three
Right.
times, but they're all valid, hopefully, right? If it's a
Right.
good model. The
Right.
same thing with peptides and proteins. We may prompt the model three times with the same, like, I want a good, non-toxic protein that binds to this virus, right? And in that case, of course, these are different objectives. We can then generate different molecules that all should be optimized for that objective. The space is very large, but of that space of possibility, this area may have the solution. And so we want to sample from that area that has the solutions to the problem that we're asking. And in this case, it's a problem that requires us to optimize multiple things. And so as that space becomes much more narrow, the
Yeah.
number of good solutions becomes much more narrow. If you ask very specific questions to
Yeah.
ChatGPT, you probably don't want 10 answers to that question. But
Right.
if you give it enough of a broad space to search, right, it can come up with and generate. It's not
Yeah.
filtering, it's generating sequences that would satisfy your objective.
I'm curious. I know you're not using ChatGPT to do this,
No, no,
but
no, no.
but does ChatGPT do a pretty good job at understanding training data on sequences of target proteins? Could you give it that?
That's a great question, and it's a very controversial question. This is
Yeah.
my answer, Brandon. Right now, the answer is no. And
Yeah.
we train domain-specific models. We train language models, but our language models are
Yes.
not trained on natural language like English. It's trained on protein sequences
Yes.
that are text. But my prediction and my forecast is that the natural language models, Gemini, ChatGPT, Claude, will be
Mhm.
good enough in the next 5 years, let's give it, where the domain expertise is so broad
Right.
that if you gave just ChatGPT the protein sequence and say, ChatGPT, please generate a protein that binds and has these properties, and then we made that molecule and we tested it in the lab, which I have a wet lab as well. I
Yeah.
think in 4 or 5 years, the model will be so robust in its domain knowledge that those molecules will work. Now, the answer right now is no. Like you ask ChatGPT to produce a peptide that, destroys protein A, and we test in the lab. People have done this, it doesn't work. It's just
Okay.
not, it doesn't have the biological and physical intuition yet. But there's no reason it can. And I think the answer is sadly for maybe people like me that train very specific and biologically understanding models, one day the open AIs, the Googles of the world will just bake it all into that single model.
So I do want to talk about kind of your testing process for the output, basically. How do you compare the current process of doing this to how this was done previously? So now we have these super robust models that you're building and training on sequences of proteins, and you can get solutions probably very quickly, potential solutions. What did the process look like 5 years ago? For example,
Sure.
with something like Wegovy, how was that created? How was that designed?
It's a great question because you asked something that when I give talks about my work, this is the first thing I said. Look at Wegovy. Wegovy was an Ozempic was discovered in the 80s before I was born, and
Oh,
it took until 2021 for that molecule
mhm.
uh, to be approved by the FDA, right?
Right,
For the purpose. What took so long, right?
right.
Is the question I ask. And the answer is that it's not even that we had GLP-1 in
Yeah.
the 80s, and it was natural. It was discovered from the biology. But what happened, Brandon, was that these molecules, even though they were made, and sometimes we don't even have them made, they took years to modify, to adjust, to optimize, to have properties that if I took it, or if you took it, or someone else took it, it would just be destroyed immediately. It would have no
Oh,
effect because it had no not good drug-like properties. And
oh.
so now, I think very recently, like in the last year, what we and a few other labs have been doing is saying, I don't just need this to bind to GLP-1 receptor, which is what GLP-1 does. It binds to another protein. I needed to also not be toxic. I also needed to have a long half-life so it doesn't get
Oh,
degraded, etc., etc. And so I think that now, in the past, it would take little tinkering. A lot of medicinal chemists would look at the molecule and be like, if I do this change, test it. If I do this change, test it in the lab until we
right.
found a molecule that would pass an FDA trial.
Okay.
Now we can immediately be like, oh, here's a model. I'm going to give it GLP-1 receptor and I'm going to tell it, make me a molecule that fits all the good drug-like properties. It generates a small subset of those possible molecules. We go into the lab. I have a wet lab. We synthesize that peptide. We put it into an animal and we see that it has a
Oh,
good property. That now is a month-long process. Well, in
okay.
the GLP-1 case was a 20-year process.
That's crazy.
It's not just the speed of making a protein or a peptide. That, you can argue that DeepMind with AlphaFold and David Baker with all their models, sure, they can develop a protein that binds to GLP-1R. That's not a problem. The problem is it going to make it through a clinical trial? And I
Yeah.
think that the answer is we're now getting good at baking that in to our generative models. That's what my lab focuses on,
I
such
see.
that it has a high chance of not only working in the lab, but working in a patient in phase 1 and phase 2 and phase 3.
So it's the process of translating it into a drug that we could take. That time has been shrunk by
That
years.
time is going, yes. And I think it's still yet to be 100% proven because we
Yeah.
don't really have an AI designed molecule yet approved. But I think that that is the promise of what this field is going to provide for a society, is like not just for drugs. This can be for anything. This could also be for, like, we develop peptides and proteins that bind to heavy metals like lead. So just the developability process of the actual product, right, which in
Right.
this case is the molecule, is shortened because we can incorporate those objectives into our designs.
Okay, so when you test this in your lab, you come up with a solution.
Sure.
Okay, now you're going to test it, whatever the
Yep.
process looks like. Is this difficult to do? And what is the accuracy like now for these new models
Yeah.
or these new, I guess, potential proteins and peptides that are being returned by your models? Like, what is the accuracy? Is it very high?
It's higher than what it would be. I think that if you think about what the accuracy was a few years ago before AI kind of came into the fold, it
Yeah.
could have been 0.0000001%, meaning a pharmaceutical company would have to screen for every new disease 1 billion molecules just to hopefully find one that is semi-good, and then they optimize it from there. That's how drug discovery worked.
Yeah.
Now, if you give me like say in the last 2 years, if you now give me a protein that causes a cancer and you say, make a drug to it,
Yeah.
I could at least give you a good starting point, meaning I will make you something that binds to
Oh,
that protein in about 40% with about a 40% hit rate, meaning I'll get you a starting molecule.
wow.
Now, the answer is, what is more important? Is it the number, the 40%, or is it that of the 40%, there's at least one that will get through a clinical trial, right? And so I think the answer is we'll stay at that 40%, which is more than enough because testing 4 things out of 10 is like super trivial for even an academic lab like mine. But more importantly is, will those 4 out of 10 be things that pass clinical trials, right? So we have more shots on goal, Brandon, right? Because we have more higher success rate. But more importantly, we also have a higher confidence that those will not just like randomly like kill the animal or
Right.
randomly like, cause liver tox in a patient at phase 1,
Right.
right? So I think that improvement in confidence at every step is what's going to be really important to ensure. So the accuracy is good, but the
Yeah.
accuracy at every stage of clinical development is going to be something that this is going to hopefully accelerate very fast.
How fast for you to create a protein that binds to a given new cancer? Like you said, here's a new cancer. Can you create something that binds, that gets us started, if you will? Is that minutes?
Minutes, seconds. I mean, the model
Yeah.
will make the molecule for me to then take the molecule, put it into, cells, put it into an animal. By cells, we'll probably have good validation in a week, and then in animals, like in 1 to 2 months, right? But again, like what I'm saying is that can be all amazing, right? That can
Right.
all work well. But if you go to a human and fails, it's just
Sure.
because it is bad, that would be a different thing. So we're trying to make sure that all of it settled at that first 1 minute of design because these models are really fast.
Yeah.
Then everything else is just plug and play. And that would be nice, right? Because I can tell you, cancer and all these diseases, yes, we will have a good shot at them with these types of models. More importantly, though, in terms of diseases like viruses or toxins or biological weapons that could happen in the next, I don't know, like 10 years, where people are using AI to design like very pathogenic viruses. That could be really scary. We need a way to quickly develop the antidote to those and
Yeah.
deploy them fast. So that's where speed really matters, and that's where we're going to be able to do it.
Yeah. Wow, it's interesting. I can't help but equate it to like AI is now being used to create computer viruses, and the AI is more sophisticated to be able to then
Counter.
prevent or remove those
And
viruses.
I think that's the answer here, is that not only are we fighting these cancers that have been around forever and these diseases that
Right.
are there, but we're also fighting new things that could be created with the pretty impressive tools. Like I told you,
Yeah.
protein design doesn't just have to make like a gene editing protein. It
Right.
could also make the most, why SARS-CoV-2 was so successful as a virus because it was just the balance of infectivity and pathogenicity. People didn't die immediately, so they could pass it on. While MERS, you just died immediately, or like SARS-CoV-1, you just died immediately, so you couldn't pass it to everyone in the world. But there will be people, unfortunately, that will use protein design to make super pathogenic viruses and toxins, and
Right.
we have to be super prepared to design the antidote quickly and make sure that if a patient took that immediately, they would be not dying from the actual molecule itself.
But it's crazy. It's like you're using the same technology.
Yeah,
You're
it's the same technique, right? Protein design is that powerful, and we're going to
yeah.
have to be very ready to use it in a good way, let's say.
What kinds of problems can these AI designed proteins help solve outside of living systems? Like, for
Yeah.
example, in the environment. Even though the
Yes.
environment is made up of living systems, what other kinds of problems can it help
Yeah.
solve?
So I think we're working on a lot of those. I mean, of course, I like biotherapeutics as a great starting point for us to try out our systems and we can cure diseases, which is
Yeah.
super important. But we're working, I mean, we have a really exciting work on heavy metal binding proteins because heavy metals contaminate a lot of environments. I'm talking about lead toxicity outside of Philadelphia is a huge
Mm,
problem. Cadmium, copper, all this silicon waste that is being produced from chip manufacturing.
okay.
So that's one thing that we're able to design peptides and proteins that sequester and remove these heavy metals.
Mm,
PFAS, right? Microplastics. People are developing enzymes that are able to destroy and degrade plastics that are polluting our environment. These are called Peptides enzymes. So those are some examples. I think that we're really interested in not only being able to sequester bad things, like kind of sweep them up, like metals and plastics and toxins and etc., but
right.
also degrade them, like not just like remove them, but like destroy them. And so that requires both binder design and enzyme
Mm.
design to
Oh, I see.
come together to make these molecules that will hopefully help our environment. There are also folks who are doing like photosynthetic enzymes for better crop yields, right? For, healthier agriculture. So I can really name you many applications, but just as many as there are in the therapeutic space, there are in environment, agriculture, food security, and and even climate change, right? Biofuels, if we have very good enzymes that can catalyze biofuel and bioethanol production in microorganisms, that could be a nice alternative source of energy as opposed to just using oil, solar, or wind.
And then what is the testing process for these use cases? Like, is it very similar? You come up with potential solutions, we're going to test it, see how it works.
Yeah.
And then what's the approval process? Like
Yeah, the
for a
approval.
living system, it's very complicated, right? You're
Yeah.
dealing with eventually patients, humans. What is the approval process for these types of that end up as products?
Exactly. I think it's just like you are working with the FDA on clinical
Yeah.
trials. You will work with the EPA and
Yeah.
even the FDA, or I guess on like more non-therapeutic opportunities as well. I think that the good thing is that we don't have to deal with like the safety issues there, but more importantly, it's there from are we damaging the environment? And so
Oh,
for
right.
us,
Okay.
I think the
Yep.
hardest thing is scale. When it becomes to in humans, I need a specific dose that I would give, say, Ozempic to you. And so I
Right.
need to be good at that amount of molecule, right? If I give you X dose and you can take it and you're healthy and happy because of it, that's great. But in the field, right? For plant bioethanol, I need to like scale it to like every freaking plant,
Yeah.
right? Or I need to scale it across an entire river. And so I think manufacturing is the big bottleneck rather than a regulatory approvals I mean, manufacturing is a bottleneck also in therapeutics because it has to be safe and not like the manufacturing process has to be clean. But here, I got to make a lot of it. And that I think
Right.
is the bigger challenge than it
Oh, right.
is just the actual design of the molecule itself.
I should have asked this earlier. How do you know or decide what proteins to design?
Yeah.
How do you decide what you're going to work on?
I think the problem is that a lot of AI folks don't really understand the biology or don't understand the actual application. They just know they're good at AI, and if you give them data, they'll train a good model. I think the
Right.
good thing about our lab, and also I've started multiple companies as well, is like hopefully everything works backwards. Here's the problem. I want to cure this rare genetic disease, or right? as the main problem. Now we
Right.
work backwards. What do I need to cure that disease? Maybe I need a peptide that would bind to the protein that caused the disease. Okay, so I need to make a peptide.
Yeah.
Now, how do I design the peptide? I need that peptide to have good therapeutic properties, and I need it to bind to that protein. So I need a model to do that. And
Right.
then you work backwards, but I don't have a model to do that. So I need to develop the theoretical framework for that. And then you go backwards to that problem. The good thing is I always listen. And they're all in Penn, there are amazing doctors who work on all different sorts of rare diseases and cancers and therapeutic problems. I talk to my friends over in environmental engineering to say,
Okay.
and so I say, where is the best ability for the tools we have, which are AI tools and these peptide protein like manufacturing capabilities to merge together and solve that problem. And so for
Right.
me, I'm looking for broad problems, but I listen to experts in those problems to see if our solution works. For me personally, we focus a lot on a subset of diseases like substance use disorder, rare pediatric cancers,
Mm.
neurodegenerative disease. I'm choosing these because one, a company won't work on them.
Right.
They may not be commercially viable. If I wanted to work on something that is commercially viable, I will start a company off of that because I need the money to do it. But in
I
the
see.
academic lab, I can choose problems that a company would not touch, but still need a solution. So my decision tree is largely dictated by what can I do and what I'm uniquely positioned to do in the space that I'm in.
What other kind of technology is required to make all of this work? You build models. First of
Yes.
all, you're building models in what? Python?
All of AI now exists in Python,
Yeah.
and the algorithms are really dense mathematically. So we definitely need innovations in math. People cannot forget it's not just about coding up a model.
Right.
Some of the models, just the current models, like a GPT-like model, may not be enough. We need to develop
Right.
new theory. I mean, you can see from the board, right? Like a lot
Yeah.
of theory being developed. Two, definitely you need the platform, the coding platform, the GPUs that will allow us to
Okay.
train on large data. But I think the one thing that gets forgotten, Brandon, is the high quality early data that the model can use and learn something meaningful.
Oh,
In
right.
natural language, if I just train on Reddit data, it would probably be pretty bad. Now that I have Reddit data, which is great to have, and I have like New York Times data, right, to train on, that together produces a really robust model. So it's the quantity and the quality of base data that I can apply a good mathematical formulation to that gives me a very good, powerful model. Then you need the ability to make the outputs that the model is producing. And that is not trivial at all. It takes, while my AI team can, take a few $100,000 to set up with our GPUs, it can take millions of dollars to set up a good lab space, good manufacturing processes, you know.
Right.
And so I think that at every stage there's different requirements, but I need them all to be able to solve the problems I do. So for me, one day can go from helping on a proof with a student to
Right.
helping another student learn how to inject an animal with a molecule. And for me, that's just a typical day, right?
Wow.
In the lab.
What is the training data? You mentioned training data. What does
Yeah,
it look like? How much data is there? And also, are you building a brand new model for every
yeah,
use case or
yeah.
problem? Do you start with a base model? We have a protein base model. Now we're going to develop on top of that. It has like the core training data, if you will.
You're speaking my language because I think everything is a transfer learning problem. I don't think we ever need to retrain any model from scratch. Here's
Right.
how we think about it. So remember, we're trying to generate things. So we have a base generative model that maybe can make you a peptide or a protein. And that's pre-trained. I've trained it on
Right.
every protein peptide I have under the planet.
I
And
got it.
all it does,
Yeah.
it spits out a protein or a peptide. It doesn't, those protein peptides don't do anything specifically, but they are good proteins or peptides.
Okay.
Now, so that to train that model, Brandon, requires, I would say, millions of data points, right? The 100 million protein sequences would be enough to
Right.
train a good model. But we have those and we train them. The problem is that you don't have 100 million sequences that also have some experimental data associated with them. No
Okay.
one, I mean, that would take, trillions of dollars. So all that model does is generate proteins. Now, the frameworks that we're developing and all that are being used in the lab, we have smaller models that say, given a protein and a peptide, predict, do they bind? Given a protein, tell me its activity. Given a protein or peptide, tell
I
me
see.
its toxicity. And
Right.
those models to
Yeah.
predict properties don't require millions of data points. Those require thousands of data points.
Oh,
And
okay.
if you had just good diversity in those thousand, you could give me a new protein and I would say, yes, it binds. And then as I generate with this base generative model, I'm
Mm.
basically altering the generation path of that model to optimize
Oh,
this property. And so as you can see, it's a very plug and play model. You don't
yeah.
need to retrain this ever. You can optimize it for sure, but you really want to have good predictors that basically get you to the solution you want and have it guide, right? So guiding these. You could give
Wow.
me another today, Brandon, you could give me a predictor for, I don't know, like fluorescence, like make it green, whatever, make
Right.
the protein turn green while doing its activity. We will have a predictor of green fluorescence. Now that can come in. So the good thing is we've gone beyond this need for huge amounts of data because we
Mm.
decouple the generative process and the controllability process. We decouple those systems, or I guess we couple them, but we don't force them to be jointly trained.
So it's like the base model gives you a template.
Yeah,
Everything's going to start with the same template. It's like if I was going to write a document,
yeah,
right? I could have ChatGPT. Give me something to start with. Give me like
yes,
what I usually start with, right?
yes,
What's my typical outline? Then I take that and I build upon it. Is this kind of similar? Here's a basic protein that binds. Now
yeah.
you're going to customize it for your use case.
And it's like when you were little, we learn English, right? Like to learn or whatever language we speak.
Yeah.
That's like our base template. But as you get older and as you train your brain, you're learning to give a very good speech. You're learning like the proper sentence
The
structure,
nuances. Yeah.
the nuances, and those nuances are things that you train over time. But
Yes.
remember, your base English is your base English, right?
Right.
You can optimize that base English as you get older with some new words, some new things. But that ability to speak English is just a core model that you have in your brain. And so the specific models are these unique knowledge bases that you develop over time. And then you say, okay, now I want to write an essay, or I want to give a speech that really explains this topic. You're basically integrating these two things together.
How innovative and unique is what you're doing?
Yeah.
Like compared to like, are there like thousands of labs that are doing this?
So the framework I just told you is very like new. Like
Yeah.
this idea of like generative modeling predictors, have the predictors interface. That requires a heavy amount of math to make work really well. It's not a
Yeah.
trivial like integrate these two and yay, it works. It's like how to do that well is very tough. And so there's only a few labs I would say that have both the like knowledge of what the problem is and the theoretical capability
I
to
see.
just to implement that. Now, once that's developed, there are a lot of labs that use the tool and
Right.
apply them to different problems. And then there's more labs that can use the higher level, like APIs, or maybe even just like a GUI that would use your mod. So
Right.
I think that what we do is really unique in one, that we know the biology, we have the theory, we can take it all the way from heavy theory all the way to animals and make these problems like solve the problems. But we also know our gaps, and there are folks that are very good, that are just theoretical mathematicians that have no idea of biology, but we bring them in because we need their
Oh,
knowledge. And there are clinicians who are like
right.
experts in X disease that I've not studied X disease for 20 years or treated patients. I'm not an MD, but
Right.
those people start coming into our with us, and we together, a theorist and a clinician may be in the same team working together, and we develop those molecules. I think that is a unique capability that we have that almost no other lab, I think, in the world does. And we're lucky to be in a place like Penn that, I can find those people. I can be this person, and
Right.
we can, do really, and we can translate very well.
So, for example, you're working with clinicians at Penn Medicine on very specific diseases, use cases.
Exactly. Exactly.
They're the subject matter expert. You're
Yeah.
working with them on like what it is you're targeting.
Yes. And so, for example, like one of the diseases that we were on the Today Show like a few months ago on this rare disease called Alexander disease. The clinician that treats Alexander disease is at Penn. She's like the main
Mm,
clinician for this disease. And so for me, my goal is to talk, or what we do is we talk to Amy at Penn and say, Amy, I need a molecule that will be good for your patient, right? I need to
right.
make a molecule that will be good for your patient. She can tell me that's what she needs. And so I have to be like, what does it take to get me there?
Yeah,
Right?
right.
I need to, generate a peptide or a protein that can be dosed orally, and, the patient will take it and it'll help remove this protein. And then I'm like, wait, wait, I can do all that, but I hit this point where I'm like, I don't have the framework that would design that sequence, right, of that molecule. Now, Mole Tao or Jia Tao Gu, who's a professor at computer science at Penn, I can be like, Jia Tao, we're very good. We can go all the way back theory, but there's this really difficult question that we just can't go over the math to like get this like say
Yeah.
multi-objective framework to work. I can go to him as well, right? And so
Wow.
and so then and like who would have thought that like a theoretical computer scientist and a clinician, would be working together, and we would be working together on a problem to cure this patient at Penn. That is something that I think is we don't realize we need, and we need to
Wow.
be able to do.
That is so cool. I got chills actually when you talked about that process of like just bringing in the necessary people to just that final difficult step of whatever it is, the math, the algorithm, like bringing someone very specific in to help solve that problem.
And I think people don't realize it. Sometimes if you go that far back, right, that far into the weeds, the solutions to solve problems here somewhat rely there. I think everyone
Yeah.
thinks that we don't need to go that far back. Like pharma company can go
Yeah.
probably up to here. Med chemist can look and find them all. I'm like, no, like let's use the brilliance of mathematics
Yeah.
and algorithms to solve this very tractable, very meaningful problem. I think
Right.
people just shut their brains off when it gets too hard theoretically because they're like, it's just theory. Who needs it? But who knew that, some core mathematical principle is the key to solving and curing this rare disease in these children. People don't look that far back, but we do, and I think
Yeah.
that's the cool part of our work.
How many problems are you working on right now? How many different models, use cases are you working on right now?
Tons. I think the point is we would also love to have models that are broad enough that they could help us solve many problems, right?
Yeah.
For example, if we had a model, we have models that can make a peptide for pretty much any protein and make it very drug-like and very therapeutically
Mhm.
viable. And so one model, but we can go after 10 diseases with it. And
Yeah.
then there are models that need to be specific, like our metal binding, our heavy metal binding problem, that needs a specific model that cannot be translated
Yeah.
from a therapeutic question. And so I think we are trying to find this very nice alignment of good models that solve in many problems, and then
Yeah.
they can be applied in many directions. I don't want too problem specific models because then
Yeah.
there's no transfer, right? Like you said, it's
Yeah.
nice to have a base model that is just good
Yeah.
generally, and if I want to adapt it for X task, that would be the ideal solution.
Would you say you're working on a dozen different protein designs?
Sure. I think we have diseases from rare disease and pediatric cancers. We have collaborators who are working in larger, maybe bigger cancers. We also have, different applications like the metals and the plastics that we're working on.
Right.
But I think the thing is that where is the right place to work on some of these?
Yeah.
And this is why I've started multiple companies, because some of those problems I really need the resources, the infrastructure of a company to actually be able to solve that problem. And so
Right.
I'm on working on the fourth one, but the first few have really translated some of the technology from the lab very well.
Yeah. So I'd like to talk about your companies.
Sure.
First of all, when is the right time to start a company to translate academic research into an actual therapy or product? Why is that the right solution sometimes? And then you said other times, or you kind of implied this, you're not going to start a company necessarily to bring something to market. Like, why start a company versus not starting a company?
I think one thing I realized, and I have to, I have very idealistic when I first began, and we're like, oh, I don't need to start a company. I could just do everything academically. And then
Yeah.
you realize when you need to scale up for a clinical trial, or you need to scale up for
Okay.
field work of a heavy metal, you realize that the grant money that
Okay.
academics are capable of getting just don't get you there. And I think that is when you realize, okay, if it's a small rare disease with 3 patients, sure, I think academia makes sense because it's a small cohort and no
Mhm.
pharma company is going, no venture capitalist is going to give me $1 billion to get a drug that only treats 3 patients where they cannot make much money and return on their investment. Now,
Right.
there are a lot of systems where if we got a lot of money and we did that, the return on investment for the VCs will be large. So my answer is this, is that one, how far can I take it
I
academically?
see.
Hit that barrier because the talent is, I think, still bigger in academia than it is in industry. Because think about it, all the smart kids that will eventually go work for DeepMind, Meta, Microsoft in their
Right.
AI minds, guess where they are before they go there? They're largely getting their PhDs or at least going to college. Maybe that'll change, but they come here first. So I get that. And I tell them, instead of, you're going to one day work at DeepMind, but why before that, why not work on curing X disease in my lab, right? And so
Right.
I think that talent is good. We have the funding to work on rare disease and specific problems to a certain extent well. And so only when I need the company to take the next step, meaning I need to scale, or I need to run a
Okay.
large clinical trial, or I need the resources to clinically develop this molecule further, is when I start a company. And so the first company was very interesting, Gamida, which is in phase 3 clinical trials right now for IVF. That was an example where I started a company really to raise money to do the work academically.
Oh, I see.
And then we put the money back into my postdoc lab at Harvard with George Church.
Yeah.
We did the work with a great team, and then the CEO of the company that we brought on, Dina, who's amazing and has led the company to great heights. She then was able to take that technology that had been already developed academically, raise a Series A, and go and take the company now into phase 3. So the academic and the industry partnered up to solve this problem and really bring something to the market rel relatively efficiently. And so efficiency, scale, cost, and return on investment
Yep.
are the 4 decision factors. How fast do you want to do it? How much return can I make for people who give me money to do it? Government
Mhm.
grants don't require any ROI, but investor will.
Mhm.
Do I need to scale and manufacture? Because if I do, then you can't do it in academia. And is this a problem that we can go very far with academically? So I always love using academia as an incubator for those ideas.
One company you started called, is it called Game 2?
Gamido.
What's
Yes.
that company?
That's the IVF company. That's
That's
the
the idea.
first company. And so they're, in phase 3 trials with cells that we've generated with AI models to mature immature eggs into fertilizable oocytes for IVF.
Nice.
It's a really beautiful company.
And what's Ubiqui? Is it
Tix, yeah.
Ubiqui TX?
Yeah, ubiquitous, kind of like
Oh,
ubiquitous
Ubiquitous.
therapeutics, so it
Gotcha.
sounds like ubiquitous. It's a cool
Yeah.
little that one is working on degrading the cancer-causing proteins or disease-causing
Oh,
proteins. So
okay.
it's basically taking the models that we've developed to bind and remove these proteins. Still a kind of a smaller company, but really scaling, hopefully to more applicable settings like clinical trials very soon.
Where do you think AI will make the biggest difference in biology over the next 5 years? And is it going to come from your lab?
I hope so. I mean, the answer is I'm very confident in what my team can do.
Yeah.
I adore my students. I think they're the smartest people in the world, and I think my team is better than any other team in the world. I personally
Mhm.
think that, even though that may not be true, I believe it, and we're going to make that happen. I think the biggest discoveries in biology are going to come from this type of thought: is don't just optimize one thing, optimize many things and do it together. Because I think this idea of multi-objective optimization is something that is very lost on people who do AI and also
Mhm.
people who do biology. You cannot optimize linearly. You cannot solve a problem one step at a time. You need to have knowledge of all steps of the process
Right.
when doing one step. And if we don't do that, we're
Oh,
going to really fail because we're going to always hit a road bump and we start back over. Hit a road bump. Even if this initial step is fast with AI, we're going to keep hitting a road bump and we have to start over again. So if we can make sure we cross many road bumps early, biology, bioengineering, therapeutics design is going to be solved much, much faster. And the second thing is context. I think what beauty of AI is it can integrate context well.
yeah.
Remember image generators? It's about integrating images and text into one model so that you can prompt Dolly or a Nano Banana with a text, and it generates a really good image. And so this idea of multimodal integration of different types of data into one shared model, I think is going to be something that only AI 1 can do, and one that we will, if we do, biology will greatly benefit, because now we can understand not just the protein and the peptide, but the
Right.
DNA, but the RNA, but the cell, the tissue, the environment. If we integrate that together, we'll have a holistic look at the system and we can design better molecules.
I just thought about this. I think you may have mentioned this. Could you develop a tool that basically makes that base model that you use to generate proteins, binding proteins, a tool that just does that based on your model? Would that be easy? And then you could make that available to other
Oh, we have.
labs?
We
That's
have. If you, I'm going to put a plug in for our models. We
okay.
have Hugging Face is a website that is
Mhm.
the basically the hoster of all open source AI models, and we
Yes.
have our own Hugging Face space where you can literally go in, you can type in the protein that causes your disease, press
Oh,
enter, and it generates the molecules. We had to make a big decision, Brandon, of whether we wanted to make this open source, but recently we were like, hey,
oh.
let's just give it to people. We can solve a certain set of problems, but it'd be amazing if people use our tools. And also other AI companies and tools are making kind of following this open source mindset and allowing folks to use their models to make good molecules for, whatever purpose they're interested in.
What are some good resources for people who want to learn more about AI and biology?
Yeah, it's an amazing question. I think it's really where you want to learn, right? I don't think that you need to know the core of AI to understand AI for biology. I think the
Mhm.
best way to look is look at where where the what companies right now are utilizing AI in their workflows, right? And you probably can guess, okay, they're working on X disease, right?
Right.
And so why are they using AI to do solve that? And so I think the best way to understand it is by thinking about the application first. Is saying,
Mm,
okay, who's solving pediatric or like a lung cancer, right? Where would AI fit into that process? And
I
then you
see.
can see and you can learn about the tools that would help there. And so
Right.
I think taking a
I
top
see.
down
Okay.
approach is better than saying, let me learn about how does
Right.
regression models work and then understand biology from there. I think you should do it the other way.
So, in other words, we just kind of talk specifically about AI for protein
Yeah,
creation. That's like kind of one specific use case. There are ways of learning more about that specifically that are going to differ from some other use case.
of course. I think overall, though, the theory is getting more unified, meaning one theoretical framework, like the one we talked about.
Yeah.
The one I talked about could also be used for text generation, could be used for image
Right.
generation, could be used for anything. So I think we're unifying the theoretical frameworks in AI such that they're useful across different domains, including proteins, peptides, DNA, RNA, whatever, but also text, image, video, self-driving cars, et cetera, et cetera.
Right.
Like you can think about the world. So I think the beauty is that we are now really doing a joint integration and creating one unified framework, a set of unified frameworks that work for different applications.
Fascinating.
Yeah,
You ready for the lightning round?
sure. Let's do it.
30 seconds each.
Great. Let's
What's
go.
a scientific idea you think is really cool, even if most people don't know about it?
Yeah, I think the idea of like creating a virus, antivirals very quickly, right? Antitoxins. Here's a new toxin. Let's get rid of it. I think that's something that people need to know is something that we're able to do.
One tool or workflow you rely on every day.
Great. Like I think I really love VS Code. I've gotten very used to
Oh,
this. It's like the platform.
cool.
I think AI integration is becoming very cool there.
Yeah.
But at the end of the day, I also love really good like just like archive, right? Just a great base tool to get all the data, all the papers that you want to read about,
Oh,
and then collecting them all together.
something you're excited about outside of science.
Ooh, I think that I think that outside of science, it's good to know that like good like po policy around science, is something that we still are working on and developing
Mhm.
and really refining how we establish our kind of our regulatory systems that
Right.
would influence not just science but other other like technologies really well, right? So maybe it's a little bit
Correct.
associated with science, but I really want like public policy and like maybe political science to really catch up to the advances in technology.
What's your favorite band right now?
Ooh, I don't know. I think everyone's gonna hate me for this, but I grew up a big Taylor Swift fan. And
Oh,
so
really?
yeah, and
Okay.
so I'm like one of those Swifties, and I think she's doing a really amazing job putting great music out. So I may be the most plain vanilla person on the planet, but that is my answer, and I'm gonna stick to it.
I was just talking to my wife about this. It was interesting because I was saying like how like people don't, they don't listen to music the same way anymore, right? They listen to songs.
Yeah,
Everyone's just into like, your Spotify, Apple Music, whatever. I'm gonna listen to this song by this
yeah.
artist, then I'm gonna move to another song. She's one of the few artists these days, I think, that can get people really excited about an entire album,
Yeah, exactly.
right?
It's crazy, and like I've listened to her since she first started, and I'm like, man, the steadiness, right, of being
Yeah.
able to appeal is something that I really think about, because I think if we had that ability in science to appeal broadly with our work, then people would understand it better, right? And so
Mhm.
it's kind of like taking insights from music and other industries that may help us to communicate to society better the work that we do.
Have you seen her?
I've gone to a Taylor Swift concert before, yes, and it was amazing. But of course, would be interested in going to new ones in the
Yeah.
future.
Last question, since
Yeah.
we're talking about music, what do you think about AI for music?
So I was when I was in undergrad, I ran a pop string group. Basically, it was
Oh,
so basically for a quartet that played pop music, and
yeah.
I was the arranger of that music.
Oh,
And so I really wish that I didn't have to listen and just had to like dictate manually or on a piano the songs I heard. I think AI should be able to do this. I hear a song, I want to dictate it in this arrangement, like for these instruments,
right.
generate for me the sheet music to do so.
Oh, okay.
I think that would be a cool way of like just like transposing really well. But I do think in the future, the generative design of new music, I
Yeah.
think is something I would rather not have, because I think
Yeah.
music, art are really what makes us human. And I think AI is amazing, but I don't want it to take away our humanity. And I feel like science could be a great way for AI to be useful, but I think human-made music and art is something we still should stick to.
I like that perspective. That's what makes us human. One of the
Yeah.
things that makes us
Like,
human.
as long as it doesn't take away that. I also don't
Yeah.
like it in with math, by the way. I really love that humans have the ability to solve these proofs and extend our
Yeah.
brain. Like if we solve Navier-Stokes, if we had AI solve Navier-Stokes, I'd be really sad.
Oh, okay.
Right? Simply because I think there's certain things that I just feel like if humans can do, it really makes us proud to be who we are.
Right. Wow. Amazing. Well, thanks a lot.
Yeah. Thanks,
This was
Brandon.
this was great.
Yeah, it was so fun. Hopefully everyone listening enjoys the
Yeah.
podcast, and happy to chat anytime.
Thanks a lot. Have a good
Thank you,
holiday.
Brandon. See ya. Happy Thanksgiving.
Bye. Thanks for watching. If you like this, be sure to subscribe. More interviews and demos coming soon.